Welcome to Virology. My name is Vincent Racaniello. I'm a virologist. I study viruses, and I'm really looking forward to telling you what I know about them in this course. We live and prosper in a world full of viruses, a cloud of viruses, I like to say. They infect every living thing on Earth. Every living thing that you see, besides yourselves, can be infected with a virus. And in some cases, we haven't identified the virus yet, but you can be assured that there's a virus for every living thing on this planet. This room is full of them. The air is teeming with viruses right now. It would be great to sample it. Very interesting, because all of you are exhaling and inhaling air that's laden with viruses. We eat and breathe billions of viruses on a regular basis, and we carry viral genomes as part of our genetic material. About 8% of our DNA is viral. So I want to impress upon you the ubiquity and the numbers of viruses, because it's not something you might appreciate. They are everywhere, and there are a lot of them. Wherever you go, any surface, especially common surfaces like doorknobs, telephones, buttons of various sorts, are all full of viruses. The numbers of viruses on Earth is staggering. So let me give you an example. In the water supply of the world, or the waters of the world, it's estimated that there's, say, 10 to the 30th or 10 to the 31st virus particles. It's just a huge number. It's almost incomprehensible. You can't really wrap around it. So let me give you a couple of extrapolations. If you figure how much each virus weighs, here we're looking at bacteriophages, which infect bacteria. You can calculate that 10 to the 30th viruses exceeds the biomass of elephants on Earth by more than 1,000-fold. That's amazing, because these are things you can't see, yet their total mass is more than 1,000-fold that of elephants. If you lined up these viruses, these 10 to the 30th viruses, head to tail, they would stretch 100 million light years. That's amazing, right? Again, these are things you can't see. 100 million light years is huge. Many people have done these calculations, by the way. The closest galaxy, Andromeda, is just 2.5 million light years away. So this line of viruses would go beyond that. So I'm telling you this to emphasize how many viruses are on this planet. That is part of the viral strategy, as you'll see, to make lots of numbers. So again, I said every living thing on Earth is infected with viruses. And if we just stop there, that would be a lot of viruses. But each living thing produces huge quantities of viruses, part of the reproductive strategy, as we will see. Let's take an example of one mammal that is infected with a virus. This, of course, is a whale, and they are often infected with these small RNA-containing viruses called caliciviruses. These viruses can make the whales sick. They can excrete. One whale can excrete 10 to the 13th virus particles per day into the ocean per day. One whale. And there are a bunch of these whales around there. It can make the whale sick. It can make people sick as well. In fact, a relative of these viruses is known as the norovirus, or the cruise ship virus. Often gives people gastroenteritis in closed settings, and we'll talk a bit about those. But just one example of the huge amount of viruses produced in an organism that are shed into the environment. At one time, the oceans were thought to be pretty much devoid of most life, in fact, except for mammals and fishes. But now we know that not only are there huge numbers of prokaryotes and eukaryotes in the ocean, but viruses as well. And when I was a student, no one ever looked in the ocean for any virus, and now it's a common thing. So if you look at the biomass in the oceans alone of the world, and it's divided according to prokaryotes, the yellow, protists are red, and viruses are blue. So on the basis of biomass, they're mostly protists. But if you look at numbers of particles, bacteria, protists, the viruses rule. They constitute 94% of all the particulate matter in the oceans. And every second, there are something like 10 to the 20th viral infections going on in the ocean. They are a huge source of carbon. They release carbon from the bacteria and protists that they infect, and this carbon is released to go into the geothermal cycles of the Earth, something that we didn't appreciate at all 10 years ago. So they are major players in the Earth's ecology. There are 10 to the 16th HIV genomes on the planet right now, human immunodeficiency virus. And this is calculated based on the number of people, over millions of people infected with the virus, and how many genomes each person has on average. This is also a very big number. In fact, it is so big, what it means is that out there in that pool of genomes, there is a genome that is resistant to all of the antivirals we have today against HIV. We have something like over 30 different antiviral drugs that we can use to treat HIV. And there's already resistance out there to every one of them in those 10 to the 16th genome. In fact, there's probably resistance to any antiviral that we can ever make. That's how big that number is. How infected are you? I guarantee you that each of you has at least 2 of these herpes viruses. At least 2. We have herpes simplex type 1, the cold sore virus, type 2, varicella zoster virus, human cytomegalovirus, Epstein-Barr virus, mononucleosis, and HHV, human herpes virus 6, 7, and 8. So you all have at least 2. This is based on population studies that have been done in many parts of the world. And with these viruses, of course, once you get infected, and all of you were probably infected at a very young age, you're infected for life. Those genomes stay with you, and periodically they reactivate and produce virions, and that's how other people are infected. And we will talk about those in the lecture on persistence. You each have about a dozen viruses in you at any given time. Probably a little bit more, but on average, about a dozen. People have done sequencing of blood mainly and a few other tissues of people, and they've found evidence that we're carrying around quite a few viruses, but most of us are okay. And so that brings up the question of whether any of these are beneficial or not, and that's really something we don't know the answer to. Now, this is a map of surfaces of the human body that are being studied to figure out what kind of microbiome is located there. As you know, we have more bacteria in us than human cells. And in our gut, of course, there's this huge microbiome that helps us in many ways, but also on various parts of our skin. There are very unique microbiomes to different parts of our skin. So you can see each of these parts is labeled, and the little pie chart is really showing you a distribution of different bacterial taxa at these locations. So you can see they're quite diverse. The microbiome gets a lot of attention. There's a NIH microbiome project to figure out the microbiome at all these body sites and what they're doing, but the human virome is being pretty much ignored. Yet we know there are viruses at each of these sites, and there are plenty of viruses in our gut as well. We all have viruses in our intestines, in our lungs, in different parts of our bodies. And as I said, many of them stay with us for our lifetimes. I think that means, like the bacteria that are symbionts with us, they are beneficial. This is a very hard experiment to do in people, to ask whether a virus is beneficial to us or not. We can really only do observational studies where we look at people who are healthy or ill and see what kinds of viruses they have in them. With bacteria, we can manipulate the bacterial populations to a certain extent and see the effect on various human diseases. But I think in the coming years, we'll see more emphasis on understanding how viruses can help us. There's some evidence that in a mouse model, for example, a herpes virus infection of mice makes the mice more resistant to certain bacterial infections. And again, you can't manipulate people in the same way to get that kind of information. This is a pie chart of our genetic material, our DNA, showing what fraction is devoted to various things. And you see the protein coding genes are quite small, 1.5% of all our DNA. But what I want you to show you here is this 8.3% labeled LTR retrotransposons. Now, what that is will become very clear to you in about 7 lectures or so. but this 8% of your genome is viral. It is derived from retroviruses, and these are retroviral sequences that infected us a long time ago, probably before we were Homo sapiens, and have stayed with us ever since. They're integrated in the germline. They are passed from parent to offspring. And they are there. It's a big fraction of DNA. At least a few of these genes, these viral genes, contribute to us. We know this for sure. But what are the other ones doing? Why have they been with us since before we evolved? And again, are they helping us? Very difficult experiment to do, yet I think their presence there must mean something. Now, I've spent a long time telling you that there are lots of viruses everywhere. You are constantly assaulted with viruses. Of course, you don't know it because they're so tiny and you have no sense of it. And most of the time, you don't get sick. Every now and then, you become ill. You get a cold or influenza or something else. But most of the time, you're okay in the face of this onslaught of viruses infecting you. Why is that? Well, it's because of this great immune system that has evolved. This innate and adaptive immune system, and many of you know about this in great detail, having just taken Dr. Moshewitz's immunology course. This immune system is what keeps these viruses at bay. So the constant onslaught of viruses that you're inhaling with every breath, 6 liters inhaling with every breath, for every minute of breathing, you're bringing in viruses. Your immune system is taking care of them. And we'll talk a little bit about how viruses interact with that immune system later on. Now, if your immune system is down, say you're getting an organ transplant, you have to be immunosuppressed. You are taking drugs to suppress your immune responses. Then an innocuous virus infection can be fatal. And these are big problems in transplant patients, especially those herpes viruses that are in many of us for life. If you're immunosuppressed, those come to life and they can cause serious disease. Another form of immunosuppression is through infection with HIV-1. The cardinal appearance of HIV-1 infection is immunosuppression, destruction of the immune system. And again, the simplest infection becomes fatal. So these examples illustrate how important this immune system is in keeping things in check. So obviously there has to be a balance between the virus and the immune system. The virus has evolved to be successful, so it has to overcome your immune system sometimes, but not all the time. And we'll get into some of these nuances a bit later. Now, not all viruses are bad. That may be one of the main things I want you to take away from this course. You have probably come here thinking of viruses as pathogens, and that's appropriate because most of the publicity they get is bad publicity. But many of them don't do anything, and as I said before, and I'll say many more times, I think they're beneficial. So here's an example of a virus. It's on the upper left here. It's a small DNA virus called the polyomavirus. And these viruses infect all of us at or near birth. We acquire them from our mothers, either during birth or shortly afterwards because there is close contact with our mother. So the genotype of the virus tracks very closely within families. So the mother will give it to their children, and those children will give it to their children, and so forth. And these viruses apparently stay with us for our lifetime. So at any given time, you can take a little blood from someone and ask, what kind of genotype of this virus do we have? And people have done this, and in fact used it to track migration of populations, because again, these viruses track with offspring. So here is a map showing you the migration of humans from their origin, Homo sapiens, from their origin in Africa. And the dotted line is the movement of people based on the DNA sequence that has been determined for a number of different peoples. And you can see they moved from Africa and split off into Europe, and then went into Asia, and eventually into North America. You can do the same kind of tracking by looking for these viral sequences in people, and you can track the migrations of people based on this polyomavirus, and you can see that it pretty much tracks along with the gene sequence with some differences, and in particular there is a branch here going down into Australia, which is not picked up by the gene sequencing. So these viruses are with us from birth. They stay within populations, and they can be useful. And again, I think they must be doing some benefit, otherwise we'd get rid of them. Now, in experimental systems where you can manipulate the hosts, we have clear evidence that viruses are good. And this is a really neat example I want to tell you. It's in a grass called Dicanthelium languinosum. And this grass is unusual in that it can grow at really high temperatures. So it can grow 55 degrees centigrade. And you find these grasses around the hot springs at places like Yellowstone National Park. All right, so the grass is resistant to heat. It turns out that the reason it's resistant to heat is because it has within it a symbiont, a fungus. And that fungus is shown here, Curvularia protuberata. You can manipulate this system. You can take the fungus out of the plant, put the plant at 55, and show that the plant will not grow without the fungus. Now, it turns out that that fungus has within it a virus, CTHTV. That virus is essential for whatever it is the fungus is doing to allow the plant to grow at 55 degrees. So you can take the virus out of the fungus and put the fungus in the plant, and when you put the plant at 55 degrees, it dies. So you need both the fungus and the virus. So it's a really interesting mutualism. Everybody wins. The virus gets a place to live, the fungus gets a place to live, and the grass grows where it wants to grow. So we know this because we can manipulate the system. Right? We cannot really do this in people. We cannot take out viruses and add them, so we're limited to what we can do. We can only infer. So that was just a little bit of an introduction to tell you how amazing viruses are. And this course is going to tell you how and why they are amazing. And again, I can't tell you how much I'm amazed by these viruses and how they work. In my 30-plus years of working with them in the laboratory, I've only gotten to become more and more amazed as time goes on. And I hope to convey some of that enthusiasm to you. So I want just a few words on what my goals are for this course. This course is a little different from most virology courses in that it's designed to give you a big picture. I'm not interested in giving you a lecture on influenza and measles and polio, because as a first virology course, this would not be useful to you. You would not learn any principles. You would not have an overview of how viruses work. That would be a great advanced course, and I might do that someday. But for this course, I want to give you the big picture of virology, how these viruses work, how they get into cells, multiply, and cause disease. So virology is an integrative discipline. It's not just the virus, but it's the virus, the cell that it infects, the organism that it multiplies in and spreads from organism to organism. So it involves many different kinds of science. It's not all about learning what the virus looks like, what kind of disease, and what kinds of genes are involved. It's about learning the whole picture. So that's why I want to treat this as an integrative science, so you get a picture of the whole thing from beginning to end. And this course is divided roughly into two parts. We'll spend the first 13 lectures or so going into the molecular basics of how viruses are built and how they work, how they make more of themselves. And then the second half, we'll talk about how they get into a host, spread, cause disease, and how the immune response responds. Now, the last point is very important, and I think most people are frightened by viruses because they don't really understand them. And I think the press takes advantage of this by scaring people every time there's an influenza outbreak or a new virus that's emerged from a bat reservoir. The headlines are meant to scare people, and people get scared because they don't really understand what's involved. And I want you to leave here in 13 weeks knowing more about viruses than 99.9% of the world's population. I think that will help you in the rest of your life, whether it's to make a decision about a vaccine or to understand why your children have certain symptoms when they have influenza, or to answer a friend's question about why this virus is making you sick. I want you to have that information, and it will make you better members of society. A couple of examples of the virus is in the press. So I have sort of a running argument with journalists who report on viruses because I think they get it wrong often. They don't think about what they're saying. And of course, they haven't taken a virology course, so they really don't know what they're talking about. There are certainly many good science writers out there, but here is an example from a CNN broadcast from 2009, and please don't get your science from CNN. This was 2009, a brand new strain of influenza virus emerged, H1N1, and a study was done in ferrets to understand how this virus behaved. And what this gentleman is reporting here: can ravage the lungs, spreads through respiratory systems, causes lesions, doesn't stay in the head like seasonal flu. All this is done in ferrets, but they failed to mention this. It's just an aside for them because it's not news. Well, it turned out that the studies done in ferrets were wrong, and in people this virus was quite mild. So again, this is an attempt to make something sensational, but not really understanding what's going on. See here, swine flu is different. Study H1N1 can ravage lungs. I don't think they even mentioned ferret in this broadcast. So this is the kind of thing that I object to, and I see it every day, and I will often write on my blog about it because I think in a way it's my goal to try and correct some of these errors. So if some of you one day do journalism of some sort, you will have a background in virology that will serve you really well. Here's another example of an editorial published in 2012 in the New York Times. And we will talk about the story surrounding this editorial in one of the last lectures of this course. This concerns avian influenza virus, H5N1. And a number of experiments were done on this virus, and the New York Times basically said in this editorial, which they called an engineered doomsday, it looks like this research should never have been done. This was research to make the H5N1 virus transmit among ferrets by aerosol. And the New York Times and many other people assumed that because these viruses could transmit among ferrets by aerosol, that they would kill half the world's population if they were released from the laboratory. A conclusion which has absolutely no experimental basis, and which in fact is wrong. If they had read the paper, they would never have written this. In fact, this editorial was published before the scientific paper came out in the literature. So I don't know where they got this from. So this is nonsense, utter nonsense. And again, when you read this, in 10 years, you're out there on a Sunday drinking your coffee, reading the Sunday Times, and you read something, you'll think back, Racaniello said to be careful of these journalists, and you'll remember the right thing. So this is part of my goal in teaching you virology. Let's define a virus now. This is a very important definition because this embodies everything that viruses do, and it can really help you to understand exactly what a virus is. An infectious, obligate, intracellular parasite. Okay, let's stop at that point. It's infectious, meaning it can go from host to host. Obligate intracellular parasite. It has to go inside of a cell. And it's a parasite, which means it's one species living in close association with another and benefiting at the expense of that other species. All right, so implied in this is badness, but as I just finished telling you, they may not all be parasites. Comprising genetic material, DNA or RNA. So as we will see, viruses can have DNA or RNA, and they are surrounded by a protein coat and or an envelope derived from a host cell membrane. So you see here pictures of various different sorts of viruses, and we will go through some of these in this course. Here, for example, is one where the nucleic acid is surrounded by a protein coat. Here's one where we have both a protein coat and a lipid bilayer. And then there are viruses that are just composed of lipid around the nucleic acid. All right, so all those possibilities are embodied in this definition. Okay, so this is really important, an obligate intracellular parasite. An interesting question is whether or not viruses are alive. And a while ago, I put a poll up on my blog, and we had almost 3,300 responses. And it's really interesting. They're divided between yes, no, something in between, and then a little bit of I don't know. Unfortunately, Survey.com took this away from me a while ago, so I put up a new one just last week, and we don't have that many responses. But I think we have 100 or so, and again, it's divided among these first three possibilities. So just for the fun of it, if you have an opinion, you should check it out there. What do I think? So after many years of thinking about this and listening to people argue and discuss it, I do think it's an interesting discussion point. The answer may not be so important, but when you discuss it, it makes you understand exactly what a virus is. My view is that a virus is an organism with two phases. So virus, the word virus, means an organism. And the two phases are, on the left, a virion. A virion is the particle, the virus particle. It's not the same as virus. Virus is an organism. The virion is not living. It's infectious. It can infect a cell, but if there are virions sitting on this table, for example, they will do nothing. They will not reproduce. They have to get inside of a cell because they're obligate intracellular parasites. So the virion is not living. However, once the virion gets into a cell and infects it and goes through its replicative cycle, which we will talk about here in a couple of lectures, the cell, of course, is living, and because the virus has taken it over, you can assume that the infected cell is living as well. So I think this is a reasonable answer to the question. So if the question is a virus living, the answer is yes, because a virus is the organism. But if you get more specific, if you say, are virions living, then I think the answer is no. Now, my question on the website is cheating a bit because not everyone has heard this argument. And in fact, if you ask 10 virologists, not all of them would agree with me either. But I've become comfortable with this. You don't have to believe this, but it's something worth thinking about. Now, as I said before, every living thing on the planet has a virus that infects us. And so when you study viruses, you're learning about the cell. We can learn about individual cells. We could learn about individual hosts as well. You cannot do much with viruses outside of a cell. You could solve their structure, their three-dimensional structure, but that only gets you so far. You can't really understand them. You have to look at infected cells. And as a consequence, what you do is you end up learning about the cell. And so many discoveries have been made about cells using viruses as probes because they are intimately involved with cell machinery. So for example, splicing of messenger RNA was discovered in a viral system. What we know about DNA replication, origins of replication and how they work, mainly sorted out initially by viral system. The cap on messenger RNA discovered in virus-infected cells. Just a couple of examples of that. So because viruses need cells in which to replicate, you always learn something about the cell when you study them. Now here's one of my pet peeves. I have a few of them, as you'll see, and that is try not to anthropomorphize viruses. They are inanimate objects. They don't think, they don't hope, they don't wish, they don't attempt. They don't do any of these human-related activities. It's very easy to do this. In fact, if you listen to other virology lectures or read other virology textbooks, you will find viruses attempting to do something or evading. We think this is wrong, and it's not just because we don't like it. There's a reason behind it. If you make viruses human-like, you are assuming that they have direction in their activity, that they're doing something on purpose, that they're responding consciously, but they're not. Viruses are passive vehicles. They have evolved to respond to environmental cues, what's going on inside of a cell, and they're simply executing chemical reactions. So if you don't anthropomorphize them, you will better remember that they really are passive vehicles. So in my textbook, we went through very carefully and removed all of these incidences of making viruses human-like. So I would like you to do the same thing. Try and avoid them. I think it will help you understand viruses a little better. All right, so we define viruses. Another feature of viruses is that they're very small. And here are some pictures to give you a sense of scale. So here's an E. coli at 100,000x, and there's a bacterial virus infecting it. So you get a sense of the size of the virus with respect to E. coli. And up here is a tobacco mosaic virus, this linear structure, which we'll talk about in a moment. This is HIV-1. And then on the right is a panel of both molecules and viruses, and it's expanded here. So these components are very much smaller than HIV and all the other things on the left. So here are the expanded materials. Here's a rhinovirus, which is about the size of a ribosome, which is shown here. Here's a carbon molecule here. This is a tRNA, an antibody, and some cytoskeletal components from cells. So this gives you a sense of the size of viruses. They are quite small, but there is a big range among viruses as you will see. Here's another view to give you a sense of size. This is a cell right here on the left with its nucleus and other components. And at the top right are two different kinds of viruses, a herpes virus, the bigger of the two, and a poliovirus. And we've expanded this on the right here so you can see the viruses. So polio is about 30 nanometers in diameter. Herpes virus, 200 nanometers in diameter. So quite a big range of sizes. Herpes is not the biggest virus by far. We'll talk about that in a moment. But you can see that these are rather small compared with the cell. For the most part, viruses are not visible in the light microscope. You need to have an electron microscope to see them, but there are some exceptions. How many viruses can you put on the head of a pin? The age-old question. Well, there's a really nice website where they actually animate this. Here's the head of a pin, and in this box are viruses and bacteria. That critter there, I believe, is a dust mite. And if we expand this boxed area to the right, here are red blood cells. I think this is a pollen grain, this yellow one. And one of these is a lymphocyte. These are bacteria, the green and the yellow. And here, you can barely see them, are the viruses. And it turns out that for rhinovirus, which is about 30 nanometers in diameter, you could put 500 million of them on a pinhead. So I like to say every time you sneeze, if you have a common cold, you fire an arsenal that can infect many thousands of people. At one time, we thought all viruses were pretty small, but some discoveries in the past few years have changed all of that. And this is a wonderful picture from this magazine cover, the American Scientist, and it shows rhinovirus, which I've just told you 500 million of them fit on the head of a pin. It's pretty tiny, 30 nanometers. HIV is a bit bigger in diameter, but look at this virus. This is the Mimi virus, which was discovered not too long ago in France. Is 750 nanometers in diameter. And at the time, it was the biggest virus known. On the right is an electron micrograph of an infected cell showing two Mimi viruses in vesicles in the cell. And here is what was at the time the largest known virus. This is a virus of paramecium. But you can see the Mimi virus dwarfs that. So these are quite big. The implications are important. We'll get to that in a minute. Just last year, a brand new set of viruses was discovered called Pandora viruses. On the left is a photograph taken with a light microscope. These are the Pandora virus particles. They're about 1 micron in length, and you can see an electron micrograph on the right. They're 1 micron in the long dimension. The biggest particles, virus particles ever discovered. They have genomes of 2.5 million base pairs, and you can see them in the light microscope. It's just amazing. Okay, another feature of viruses that distinguishes them. So we've talked about intracellular parasitism, we've talked about size. Okay, viruses replicate by assembling preformed components. They're not like bacteria. Bacteria, you take one bacterium, you put it in broth, it divides, you get 2, those 2 make 4, 4 get 8, and so forth. Binary fission. Viruses don't do that. They get into a cell. So here's the trace of a virus getting into a cell with time and the number of infectious particles made. This is called a growth curve. We'll talk about this more detail next time. When you start the infection, you don't see any infectivity in the cell. It's called the eclipse period because the nucleic acid has come out of the particle and it's being translated into protein to make the building blocks to make new virions. So there's this eclipse period during which there's no infectivity, the viruses are being built, and then at some point you can detect infectivity and viruses are released. All right, so important difference between bacteria and viruses. Assembly of preformed components versus binary fission. And we will talk about this assembly a bit later and tell you exactly how it works. How old are viruses? We can look at the evolution of, for example, herpes viruses. We can sequence the DNAs of various herpes viruses and calculate what we call a molecular clock, the rate of evolution per year of the virus. And if you use that clock to work backwards, you can put herpes viruses among the dinosaurs, whatever 150, 200 million years ago that was. And there are other ways that we can say that viruses are hundreds of millions of years old as well. We'll talk about those in a separate lecture. But it's likely that they actually arose along with the first cells. Viruses probably evolved along with the first cells, which would be 3 to 3.5 billion years ago. So I think, and many others think, that viruses originated with cellular life a few billion years ago. And we'll talk a little bit about that in the evolution lecture. But the point is, they're very old. As soon as we began to record history, humans, we see evidence of viruses. So on the left, a vase from 700 BC is inscripted with, "Here this firebrand rabid Hector leads the charge." Rabid Hector, the first known incidence of rabies. And on the right, a Egyptian carving from 15 to 1300 BC. This priest looks like he has polio. The way his leg is hanging is exactly what polio does to you, and this has been cited as the first record of poliovirus. We know that in the 11th century, people tried to prevent what were virus infections, even though they didn't know they were viruses, in a variety of ways, and one of them was smallpox. It was noted very early on, smallpox was a disease that ravaged humanity from the start. It was noticed that the survivors never got sick again. People were smart, even way back then. So what they would do is take the material from a lesion of someone with smallpox and inoculate someone else with that. They had no concept of infection, but they just knew that if you got the disease, you would not get it again. So they figured we could give you the disease by giving you some extract of a lesion. That was called variolation. Not a great practice because smallpox has a 30% fatality rate, right? Not a vaccine the FDA would like, but it was among the first efforts to prevent infection. In fact, many years later, the wife of the English ambassador to Turkey, Lady Montagu, learned about variolation in Turkey, and she brought this practice back to England. In the 1790s, as we will talk about later, Edward Jenner established this practice of variolation with the same disease but using a different virus that didn't kill its hosts. So the concept of something very small, a microorganism, really escaped humanity for the first 1700 years of recorded history. And it wasn't until Leeuwenhoek in the late 1600s made a microscope did we realize that there were things out there that were alive that we couldn't see. We assumed that everything we could see was all that was living on the planet. But he made this interesting microscope, and he looked in all sorts of materials and found little things swimming around, bacteria and protists and so forth. So the first time seeing submicroscopic life. And among those were bacteria. He couldn't see viruses, of course, but it was something very small. Pasteur in the 1800s said there are things called bacteria, and they multiply in broth, and they do cool things like help us to make cheese and wine and yogurt and so forth. So he established this idea that bacteria are very small and that they're living organisms. And then Edward Koch, a bit later, said these bacteria, some of them anyway, can cause disease. So this whole, in a very short time, we had the development of this theory of microorganisms and the fact that they can cause disease. Now, where do viruses fit in? This is the first viral disease that was studied at the end of the 1800s. And this is a tobacco leaf, and at the time tobacco was already economically a valuable crop because it was being smoked. And this is a disease called tobacco mosaic disease where the leaves get mottled. and made the tobacco unsaleable. So a lot of people were working on it. And 2 individuals figured out that whatever was causing this disease was contagious, it was infectious, and it was very small. And what they did, they would grind up the leaves and make an extract, and then try and give the disease to another plant. And if you did grind up a diseased leaf and make an extract and put it on another plant, that plant would get infected. But what they found, and that was Ivanovsky in 1892 and Beijerinck in 1898, if you filtered the leaf extract through a very small filter, these were the filters that they used back then, they have very small pores in them, whatever was in the leaf causing the disease would pass through the filter. And they had very, very small pores in this filter. And so these filters would keep bacteria out. So they said, these are not bacteria causing this disease, but it's something really small. And Beijerinck called it contagium vivum fluidum. And the name virus, which is Latin for slimy liquid or poison, came to be used to describe tobacco mosaic disease agent. And after that, virus discovery began anew. There was this new entity in the microbial world, a virus. 1898, 2 individuals identified the first animal virus, which causes foot and mouth disease. This is a disease of cattle where they get lesions on the feet and the mouth, as you can see. And the key concept with this and the plant virus discovery, the agents are small. They pass through a 0.2 micron filter. But also, they would not grow in broth. So it had already been known from the work of Pasteur and Koch that if you put bacteria in broth, they will multiply. But these individuals, the foot and mouth agent, the tobacco mosaic agent, if put in a broth, would not multiply. So they were unique agents. And virus discovery after that went pretty rapidly. The first human virus in 1901, yellow fever, then rabies, smallpox virus, the first leukemia, the first cancer-causing virus, and polio, then another cancer-causing virus. And we'll talk about these viruses in some detail later. Phages, viruses that infect bacteria, and influenza virus only in 1933. Now, for many years, people didn't know what viruses looked like. Many people thought they were just liquids. And in 1930s, the electron microscope was invented. And finally, people could look at viruses, and they began to see these amazing shapes. And here is the virus of tobacco mosaic disease on the upper right, tobacco mosaic virus. This is a bacteriophage. Here is a virus related to rabies, and a virus that causes gastroenteritis. So finally, it was shown that viruses are particulate. And from that point on, people really could get a grip on what they were doing. Yes?
That list that you just showed us before,
This list?
yes.
Yeah.
Did they evolve, or was it just discovery?
This is discovery.
Okay,
Yeah.
so all these were around?
Presumably, and that's an interesting question, these had been around for many years. We just hadn't discovered them. How long is a good question, and that's something we will come back to when we talk about evolution. Yeah. And the pace now is accelerating. With new techniques, we discover more and more viruses. And in fact, that is something I'm going to tell you about in a moment. So today, this fast forward to today, we know amazing things about viruses. We have structures in 3 dimensions of many viruses solved by X-ray crystallography or by cryo-electron microscopy and tomography, so we know what they look like. For polio, we know the chemical formula because we know every protein and every bit of nucleic acid in the particle. We have enormous knowledge about viruses. And I want to convey some of that to you in this course. A few words on how we organize all these viruses. They are classified in a certain way. And these are some of the criteria that are used to classify viruses: the nucleic acid, how the shell is built, whether there's a lipid membrane or not, and also the size. Today, as far as I can tell, the only thing that is used is the nucleic acid sequence. You can now sequence a viral genome and immediately tell what family it belongs to. And if you can't put it in a family, then it's probably a brand new virus. Now, you may know that living things are classified in this hierarchical system of kingdom, phylum, class, order, family, genus, and species. The virologists only use these last 4. We have orders, which are made up of families, which are made up of genera and species. So for example, the species Zaire Ebola virus, which is shown here, is in the genus Ebola virus, and that is in the family Filoviridae. So virus families end in Viridae, and genera end in just virus. So when we talk about viruses, you'll see some of these terms thrown around. You can come back to this slide and see what they mean. Now, there is an organization that keeps track of all the known viruses. It's called the International Committee on the Taxonomy of Viruses. They have a website, and they have classified 2,618 species into 420 genera, 96 families, and 7 orders. This only represents a fraction of what's out there. These are the characterized viruses that you can grow in cells in the laboratory. Most of those 10 to the 30 particles in the ocean we can't grow. We can't culture them, so we can't study them. But this is actually a growing area of virology. Environmental virology. What's out there? As I said 10, 15 years ago, no one looked. No one looked in the oceans or anywhere. In this paper, which was published in Science, these investigators went up to Lake Limnopolar, which is a freshwater lake up down in Antarctica, and they drilled holes in the ice, and there's fresh water in there. And they take samples. They bring it back to the laboratory. They extract, they filter it to get rid of bacteria and protists. They extract the nucleic acid, and they sequence it. They determine the sequence. And from that, they can say, we recognize, polio, herpes, X virus, and we have other viruses that we don't recognize at all. And in this one study, they found 10,000 species from 12 families, some of them brand new viruses. So wherever you look, you will find new viruses simply because no one has looked before. Every environment, this is an extreme environment. If you go to hot springs, you will find viruses. If you go in all layers of the ocean, you will find viruses everywhere. So this is a really hot activity now, just to go out and sample things and look for the viruses. One of my colleagues is looking at viruses in New York City subway rats. Anything you can think of is subject. Here's another really cool study which emphasizes this idea that there are lots of unknown viruses out there. This is a study where they looked at raw sewage, raw human sewage. So they went to 3 sewage treatment plants in the US, in Spain, and in Africa. And they got 10 liters of liquid from each plant. They purified viruses from the liquid. They concentrated it down to a milliliter. And then they sequenced the nucleic acid of those viruses. And then you get lots of cellular sequences, and you throw those away. You can subtract those bioinformatically, and then you see what you have. So this panel in the middle that just shows you some of the photographs of these viruses that they took. They could find particles that look like viruses in these sewage treatment extracts. You can see various spherical viruses, those long ones, some with tails. And on the right, this pie chart shows you how the sequences mapped. So a good fraction of the sequences, the blue, 247,000 sequences, so each is an individual sequence. These are bacteria. Then they had a few fungi. This is all in sewage, remember. So bacteria, of course, makes sense coming from your microbiome. Some fungi in there as well. There's some human sequences because you're shedding cells into your feces, and so that gets picked up as well. There's some phages in there, and then there's some known viruses, 8,491 different known viruses. But look at this, unassigned, 596,000 sequences. They're unique sequences. These are probably mostly viral. And these are unknown viruses. We don't have a sequence in the database that says, yes, this is X virus or Y. So this is virus discovery. You can go out there and do this maybe for the next 10 years and still find a lot of unique sequences. The key here is that most of these are not culturable. And the techniques that allow us to find these sequences were only developed in the past 5 to 10 years. The ability to massively sequence large samples like this simply wasn't possible before. So lots of exciting stuff going on Why do we care about so many viruses? Well, viruses outnumber cellular life by at least 10 to 1. They comprise the greatest biodiversity on Earth, more than anything else. So just for that alone, I think we need to study them. As I mentioned very briefly, we'll talk about a little later, they drive global cycles, ecological cycles. And they probably have other benefits that I've alluded to as well. And finally, all these viruses out there are likely, at least in some cases, to be sources of new pathogens. So it's good to know what's out there. I think we really need to know all the viruses on the planet. So I've just told you about this huge diversity, and you're probably thinking, do I have to learn all these viruses and how they work? And the answer is, we're going to simplify it. And we're going to do that because of these two facts. So we're going to take these billions and billions of particles and put it in a way that is easy and makes sense for you. First, all genomes are molecular parasites. They have to get into a cell where they will replicate. And finally, all viruses have to make messenger RNA that can be translated by the host. They're parasites of our protein synthesis machinery. No virus encodes a protein synthesis machinery. So it has to make mRNA that the host can translate. No virus can make energy. No virus can make lipids, as far as we know. There may be one out there that we don't know about, but as far as we know. So we can use these criteria to characterize and classify all of these billions of viruses that I talked about. And that's going to be one of the goals of this course, to take an overview of all this diversity and make it simple.